Welcome back and welcome to the last lecture of today, the lecture number 9, texturization techniques, high moisture extrusion and shear cell technology. So in the last lecture, we have been discussing about we need to create plant-based meat analogs or alternative protein-based meat analogs. It's good to do that to get the desired texture, flavor and color and so on. It's also very important. However, how do we get that particular texture? How do we get that meaty texture? How do we get that fibrous texture? How do we get that juiciness? For that, we again have to process our protein ingredients together with other components. It could be water, it could be fat, it could be carbohydrate, it could be some other binders. So everything has to be mixed and reprocessed to get the texture, the meatiness, the fibrous nature of meat that we want. How to get that one? We need to again have to process. And one of the most common processing technique which is industrially employed, it's not in the laboratory scale I'm talking about, not only in the laboratory scale, industrially employed is high moisture extrusion. Okay, I'm going to talk a little bit about high moisture extrusion. Just recall my slide again. We had insect, we did not want to insect. I convert into insect protein isolate. Now by extrusion I can get different texture or fibrous nature I want, and that can be used as a meat analog, and that's where we can have insect indirectly. Okay, have you done extrusion before? Anybody here? You have done it? Maybe I personally haven't done extrusion myself, only my colleagues have done it for me. I analyzed you have also done it. But let's see that you're also welcome to share your experience if there is a question. Okay, um, extrusion is a big machine. In other words, right, where things are extruded. In other words, right, this is the basic structure of an extruder. What does it have? It has a feeding chamber, right, where you introduce your protein as an isolate or a concentrate. Of course, you are also welcome to add other ingredients together with it. It could be carbohydrate, it could be a fat, and everything will be added to here. And then basically, it comes to the second region, what we call kneading zone. Is kneading is nothing but a mixing or doing something like this, right? In that particular zone, it comes where all the ingredients that you have added will be mixed with water. Okay, water, you remember, water is important for what? Hydration, right? To improve the water holding capacity, we need to introduce water, right? It's called as high moisture extrusion because the water content can go up to 80% here. Like, if I'm not wrong, people call it as 60 to 80% will be the water content in the product. That's the reason it's called high moisture extrusion. There's also something called low moisture extrusion, where the water content will be like 10 to 40%. Okay, I'm not going to teach that today because that's not very often used for the juiciness, right? So we use high moisture extrusion. The water content in the product will be 60 to 80%. So everything will be mixed here, and then the product will be sent in this direction. We have a twin screw like this. You must have seen it in mechanical engineering laboratory, how that exactly the twin screw looks like, right? It is rotating like this one. And while it is rotating the screw, it will take all the ingredients together with, and it will be mixed at very high shear stress and pressure and also temperature. The temperature in this particular region, in these regions, can be between 50 to 180 degrees Celsius. Basically, you're cooking under high pressure, right? You're cooking the plant proteins together with ingredient in a screw at high temperature under shear stress and pressure, and that will actually change the protein structure. Now, the protein, it was originally looking like a powder, it becomes fibrous, right? And then it comes to the end of this one, there is a cooling zone where basically the temperature can go like 50, 40, something like that one, which were very, very hot will be immediately cooled down, and then the extra moisture will be evaporated there because of the condensation here. And then you get the product out, which is exactly looking like a meat analogs. But you can again have to cook it, you can again have to freeze it, you can again have to thaw it, you will also do other processing after getting. The one you get, it's not necessarily a final product, they still process it. And this is the basic principle of extrusion. Is it clear? It is one of the most commonly used method today to get the meaty, juicy, fibrous structure of meat equivalent to meat from plant proteins or insect proteins and so on. Make sense? Very simple. There is a big machine. You have a feed mixed with proteins and other ingredients that you like. They add also color and they also add some other things you want, and that will be sent in this direction, and then it's mixed with water, right? And then it will be screwed, moved in a twin screw at high pressure, shear force, and high temperature, where things will be cooked. Then the protein denaturation happens, and that will lead to the fibrous structure when you cooled it down at the end. That can be further processed, like you can do boiling, cooking, freezing, thawing, many things they do later to get more fibrous structure. Make sense? So now the most important thing for us to know, why do we get that fibrous structure? How and why? What is the mechanism here? There is nothing has been proven it actually, but there is a lot of hypothesis. There is some basic experimental studies. They postulate that these things can happen inside this machine over here. Okay, over here many chemical reaction can happen. That's what they're trying to say here. The first one is that in this particular zone here, you mix it with water, right? In this particular zone here, you mix it with water, and the temperature can be like 40, 50, 60 degrees Celsius there, right? And some proteins will have a denaturation temperature 50, but some will be usually having 80, 90, 100 and something like that. But now the protein is in contact with water, and now it's actually mixed. Coming back to our first lecture today, solubility and water holding capacity, right? Now protein is coming in contact with water molecule, the protein will be hydrated, right? Water holding capacity of the protein is increasing simply because now more and more protein is in contact with more and more water molecule, right? And then in this particular zone, in this particular zone, the temperature can be, they say, 100 degrees Celsius to 180 degrees Celsius, depending on the machine here. And that is the temperature. We all know that most of the plant proteins can also denature, right? Globular proteins usually have a very high, the albumins usually have a high melting points, melting temperature, but at 180 degrees Celsius, I can tell you that most of the proteins can be denatured. When the protein denatures, it will expose all its functional groups, right? It will expose all your functional group, and now that protein is available for several chemical reactions. Number one, it can have a protein-protein interaction. It can have protein-carbohydrate interaction. It can undergo protein oxidation. It can undergo protein-lipid interaction, because now it's exposed and everything is mixed together in one zone now. There's a huge, nice contact between the molecules, and there is a lot of opportunity for chemical reactions, right? And then that will also help because you are screwing in this way, and it helps in the stretching of the protein. At the end of the day, you cool it down. Then the open proteins, now it has undergo chemical reaction, or it can actually form these cross-linked networks, right? Now we started with the globular proteins, right? Now you got a network. Can you see? You started with the individual globular protein like this. Now they are actually a cross-linked network, and that cross-linked network will give you the fibrous structure according to the literature. I'm not very sure it is 100% true this is a chemical reaction, but most of the people believe that this is the way you get the structure. But there's a lot of other phenomena as well. Does it make sense? So you just converted a globular protein into a fibrous protein now because of the chemical cross-linking. I just talked a little bit about chemical cross-linking. Just you don't need to read this again. I'll just show you. So the protein will have three-dimensional structure like this one, and there will be intramolecular disulfide bonds, and there will be intermolecular disulfide bond between two proteins, right? When you heat it at 180 degrees Celsius-ish, right, there is a possibility that these bonds can be broken because they can undergo oxidative modification, reductive modification, and so on. Then basically you will break this intramolecular disulfide bond, then the protein will be exposed like this one. Now when there is a thiol group is exposed, and if there is another protein with a disulfide bond like this one, they can interact with each other and can form a new cross-linking like this one. So you're creating a protein-protein cross-linking, and the protein-protein cross-linking can lead to the aggregated proteins, and that can essentially give you the fibrous structure. Does that make sense? The take-home message is that in the different zones in the extruder, opening of the protein takes place, chemical reaction can take place, protein-protein interaction can take place, protein can be covalently cross-linked, and that will give you the fibrous structure. Any question here?
Sir, in case of low moisture content, this extrusion that is working on low moisture content, the dye from which this compressed material is coming out is having a major role for the product development.
Okay.
So, is there any relation or correlation between dye and high extruded product in high moisture content?
You mean the dye, this particular portion you're telling here?
Yes, sir. Sir, in case of low moisture extrusion, it's having a big role.
Yes, of course.
Because of the high pressure and low pressure outside inside it
From here to
yes
transition
it's a high pressure. When it comes to low pressure, it expands, so that makes the product fluffy, and its texture is developed in from that dye. So is there any relevant effect on in
Yes, I think one of the relevant effects I read, I honestly tell you that I never did this experiment myself. What I read was that one of the most common thing happens when the sample comes from this region to this region, they expel the water which is not actually holding on the proteins. Basically, kind of a syneresis, like it loses the water. And apparently that might have an effect on stabilizing the structure, because now you don't have a liquid kind of a jelly matrix, but you will get a matrix which is juicy, not like a yogurt. That is the one thing I know, but I am not very sure how exactly the pressure change will have an effect on final product. I have no idea, unfortunately. But what I know that they claim that when the things come from this region to this region, this is what happens. You see, the cooling. This is what happening at the dye region. They say that the stretched one, now they start cross-linking when it is cooled. I'm not very sure whether it's true chemical reaction that's happening. I don't see any very strong chemical basis for that one, but this is what people believe. I don't see a very strong evidence for this ever. Yeah.
Sir, one more point.
But do you think something will change? Because you have experience, I don't have
it. Sir, in case of low moisture extrusion, the water is having big role.
Yeah.
So when it is compressed under high pressure and high temperature, when it comes out of the dye, it expands.
Yes.
So when it expands along with the product. So
exactly.
we get cells.
Yeah.
We get the particular shape, and it depends upon the shape of the dye. Which type of product we want. So we want any customized product, we can change the dye. Similarly, I do not have worked on this high moisture extrusion. So I just want.
I'm not very sure. Sorry, I don't know how exactly the dye, its shape and its temperature have an effect. I don't know. Let's find it later.
Okay, thank you.
Good. This is how the commercial scale extrusion equipment look like. This one, they come in different sizes. Wow, huge ones are also available. We can also buy a laboratory scale of this size. Thermoscientific sells one laboratory scale extruder that you can do laboratory scale experiments. But they're also available by different companies in a very big scales. Okay. This is not something new, right? Extrusion is not something new. You know, pasta making? pasta. Do you get a shape? And in chakuli, what we call in English? Chakuli is, I don't know what is the equivalent in Hindi. Chakli. Same? Okay. They're all basically all this, it's a similar phenomenon, right? Muruk, muruk, whatever it is. Like it's basically, we don't do it with extremely high shear force there, but still we apply shear pressure, right? Still be, it's basically 3D printing according to me. Making chakuli and making all these things, all 3D prints. Now it's already there in our culture, right? But now we have machines to do that. There is another story, right? And also the noodles and all these things, they're all actually, but extrusion has a little bit high temperature and that twin screw, all these things are there. But otherwise, they all follow a similar thing. For your understanding, I'm telling, it's not something completely new, okay? And you can just see for the new people here, you can just see that how different kind of proteins produces different kinds of texture. There's a nice figure I found it from this review article. You can see that it has this chickeny structure. Can you see? Usually wheat gluten has these disulfide bonds and sulfur atoms, thiol groups that help you to get this stretching. In bread making, the dough gives you that stretching, and that can also be exploited here to get the meaty fibrous structure. It looks like a chicken breast piece. Can you see? It looks also like a chicken, but I don't think this is looking like a chicken though. And I have two other things to example here, like the problem with the color. Remember? Now, if a chicken is having this color, we make a green chicken curry in our hometown. Maybe you also make it here. But we add palak or something later. But we don't want a chicken which is already green, right? Consumer's problem. So if the protein is green, we have also problematic here, right? So that's the reason why you need to find the right ingredient. You need to remove the color from the protein to get a real this color. Okay. Now I will show you little bit about some concern. One of the biggest thing we have to remember here is that extrusion happens at high shear pressure or force, whatever it is, and the temperature, the heating can go up to 180 degrees Celsius, right? It's good. It's good for cooking. I fully agree with that. However, high temperature can also lead to some of the unwanted chemical reactions, right? High temperature can also lead to some of the unwanted chemical reaction because you have protein there, we have lipid there, you have minerals there, and they can lead to some unwanted chemical reaction, and they can form new type of chemical compounds, and nobody knows how that will affect your body. And I have received around 6.5 million Danish kroner to investigate this from the Danish, around 7 crore Indian rupees, to do a research on how exactly the chemical changes can affect the digestibility, and we're also working on how it will affect the brain. If you continuously eat extruded product, whether you will sleep more in the classroom. We never know, right? The digestibility, right? So those things we are going to, not sleeping in the classroom, we're investigating, but how it affects the memory. You learn so many things you might forget because of eating a lot of extruded product. I don't know. We are going to investigate that one. Now I'll go to the last part of today's lecture. Similar to high moisture extruder, we have another technology called shear cell technology. It's kind of somewhat new, I would say, not available everywhere. I did not see a lot of articles on this one. It does a very similar job like high moisture extrusion. It also creates an anisotropic fibrous structures using a different mechanical phenomenon. In there you have this twin screw. Here you basically have what we call shear cell. Okay. I also personally haven't seen this machine and never used myself, but I'm only teaching you based on what I have read here. Apparently it has two cones, a cup, a conical cup here, and it has another cup here on top of that one. And the sample is placed, I hope I have a diagram here exactly. So you have a cone here, the first cone, and we have another cone on top of that one, the second cone. Can you see this part, right? And your sample, the protein, carbohydrate, everything is placed in the middle. The lower cone, apparently it rotates, and then it creates a very high shear force, and this temperature can also be controlled here. Okay, you can apply high temperature, and that apparently gives you a very high fibrous structure that can be used to create meat analogs. It's a new technology. I'm not a mechanical engineer. Unfortunately, I can't teach you so much about the machines. Maybe you guys are cleverer than me. You have a bachelor's in technology, but I have a bachelor's in science. So basically, it has a 2 cups, 2 cones, and between 2 cones, the sample is placed, and one of the cones rotates, and that creates the shear stress, and that leads to the fibrous structure. And the temperature is also important here. It also goes to high temperature. Apparently, you can control the temperature here. Mechanism exactly the same that I just explained: protein denaturation, renaturation, and cross-linking. You're converting a globular protein into a fibrous structure. Okay. If you really want to know this shear cell technology and how to create the fibrous structure using protein, I really recommend you go through this video. Unfortunately, I can't append the video because of the GAN guidelines. I have put a link for you here. It's a 1 hour, 13 minutes video, but it has a lot of information about how to create a structure, meat analog structure, using plant proteins with different technologies, and what are the parameters play a role here. It's explained very detailed in here. Okay, I really recommend that you go through it when you are free. All right, and that is the class for today. So we discussed about several things today. I really cannot summarize from the morning so many things we have discussed. But in the afternoon, in this current session, we discussed about 2 important topics. How do we create meat analogs and dairy analogs? And I talked mostly about some of the challenges. I told you about it's not just proteins, it's too many other things are involved to get the final product. It's never a just protein. My lecture is about alternative proteins, but only protein alone cannot work. It has to combine with so many things to get the final food product. To get that, you need to do a lot of processing, you need to add a lot of ingredients. They have a good and bad effects. A lot of research is needed in that area. Then we talked about 2 important technologies that we use today: high moisture extrusion, and possibly in the future the shear cell technologies to get the meaty fibrous structure. We realized about the chemistry behind that one. It's protein denaturation and cross-linking. Apparently that leads to the fibrous structure, but there's a lot of other physical chemical phenomenon that we don't have to worry about. But if you're really interested more about getting the texture, go through this particular video. And that's all for today. Thank you so much for attention, and let's see you guys tomorrow. We're going to talk more about digestibility, bioavailability, and so on. Thank you, and enjoy your afternoon.